Hello, my name is Emily Wrightstone, and I will be discussing my in vitro investigation of a highly selective chemotherapeutic dye. Many chemotherapies are a race to see who survives, the patient or the cancer. A near universal motif of tumors is that they are made of uncontrollable, rapidly dividing cells. Chemotherapeutic agents which target the nuclear DNA of these cells are successful in mutating the nucleic acids to the point of cell death, therefore killing the tumor. A list of some of these commercially used agents are shown on the right. However, these drugs are not commonly selective in their cell targets beyond just the aspect of a rapidly dividing cell. This means that while they are successful in killing the uncontrollable cancer cells, the DNA of perfectly normal cells is likely to be attacked as well, leading to the collateral damage of healthy tissue. The most common healthy tissue to be affected by chemotherapeutic agents are hair follicles and white blood cells. Their deaths, among others, directly result in the harsh side effects of chemotherapies, including hair loss, nausea, extreme weight loss, and low white blood cell count, to name just a few. These low selectivity ratios, commonly around 2 to 1 or 3 to 1, are hypothesized to stem from chemotherapeutic agents attacking the nuclear DNA. There is a need for an anti-cancer agent that is more selective towards cancer cells, spares non-cancer cells, and does not significantly harm patients. Cyanide dyes have been found to exhibit strong anti-cancer properties. The figures displayed here were adapted from data found in Gilman et al. 2006, which details the cytotoxic effects of a cyanide dye family developed at Kodak in Rochester, New York. Both graphs were created by comparing the IC50 values, or the amount of compound required to kill half of the cell population, between a control and cancer cell line. These ratios were calculated by dividing the IC50 of a compound for the control cell line by the IC50 of a cancer cell line. In other words, these ratios can be thought of as how many cancer cells are killed for every one healthy cell. The control line for both datasets was CV1, a non-cancerous monkey kidney epithelial cell line. The figure on the left depicts the selectivity of a commercial chemotherapy, Adriamycin, against several human carcinoma cell lines, and compares its selectivity towards 3 members of the cyanide dye family. Even at its most selective ratio of 16, Adriamycin pales in comparison to the selectivity of the cyanide dyes, which frequent a ratio of 60 or above, even reaching as high as 300 in 2 instances of compound C. The figure on the right shows the selectivity of Adriamycin in a handful of cyanide dyes against CX1, a human colon carcinoma cell line. In this case, Adriamycin is at the negligible 1.5 ratio, while the least selective cyanide dye comes in at 60. It is abundantly clear that the cyanide dye family exhibits exceptional anti-cancer properties depending on the type of carcinoma. My research has obtained a dye from this family to use as a control compound, while we manufacture an additional one for further investigation. The dye, which we arbitrarily call D1, is hypothesized to repair a broken mitochondrial-mediated apoptosis pathway. When cells become significantly mutated or approach uncontrolled cell growth and division, internal apoptosis or programmed cell death, pathways are activated to kill the cell. This process has evolved as a method of protecting the larger organism from malfunctioning cells, which actually have the capacity to divert indispensable nutrients to favor their own growth. Cancer cells then arise when these internal cell death pathways become malfunctioning or broken, allowing these cells to continue dividing beyond their normal capacity. Based on previous work conducted on the cyanide dye family, it is suggested these dyes repair a damaged pro-apoptotic mitochondrial pathway. This pathway, as depicted in the figure here, involves a cleaved BH3 motif binding to BCL2 and consequent pore formation in the mitochondrial atom membrane. This causes the release of cytochrome C, creation of the apoptomosome, and subsequent apoptosis. Presently, the exact role of the cyanide dye in repairing this pathway is unknown. However, it is suspected to be involved in aiding pore formation in the mitochondrial membrane. Other external studies have identified that cells containing mutant BCL2 proteins were not affected by the dye. Additionally, internal computational analyses have revealed possible binding sites for D1 on BCL2 proteins, with binding affinities supporting their interactions. These dyes are potentially selective to cancer cells because the pathway is not active in normal, non-cancerous cells. 3 cell lines have been used to study D1 so far. NIH-323 is a mouse fibroblast cell line and is a non-cancerous control. A549 is a human lung adenocarcinoma line, and T24 is a human urinary bladder transitional cell line. The first major experiment was confocal microscopy in order to visualize the location of D1 with respect to the mitochondria. For this, each cell line was treated with D1 and a mitotracker green dye which was obtained from Thermo Fisher before imaging. Cytotoxicity and selectivity of the dye was analyzed through two different types of experiments, a trypan blue exclusion assay and an MTT assay. In the former, seeded cells were treated with either 0, 60, or 100 nanograms per ml of D1 and 0.09% lactic acid. Cells were then manually counted at 6, 24, 48, and 72 hours of treatment using the trypan blue to identify non-viable cells. The MTT assay was conducted using a PsiQuant MTT cell viability assay from Invitrogen. Seeded cells were also treated with either 0, 60, or 100 nanograms per ml of D1 and 0.09% lactic acid. At 24, 48, and 72 hours of treatment, the kit was used to stain viable cells with the MTT dye. After an overnight in solubilizing solution, the relative growth of each sample was then measured with a plate reader. Cytotoxicity was first calculated as a percent reduction of cells between the treatments and controls within each cell line at each time point. Selectivity was finally calculated as a ratio between cancerous and non-cancerous cells at each time point. Figures A through C here depict the results of confocal microscopy, where cells are treated with D1 and mitochakra green, the latter of which is known to localize to the mitochondria of cells. D1 holds an excitation of 514 nanometers and an emission of 575.23 nanometers, while mitochakra green has a 490 nanometer excitation and 516 nanometer emission. The D1 was added 6 hours before imaging, and mitochakra green added 30 minutes. Figure A depicts the non-cancerous mouse fibroblast NIH3T3 cell line. The light blue shading here indicates an overlap between the presence of D1 and mitochakra green dyes. In Figure C, the T24 human urinary bladder cells show a few light blue splotches to indicate dye overlap. However, the morphology of 3T3 and T24 cells make it a bit difficult to determine nuclei locations without the aid of nuclear-specific imaging dyes at this magnification. Unlike those cells, the A549 human lung adenocarcinoma cells in Figure B clearly display internal black circles. These circles reveal the nucleus location of each cell and indicate that D1 does not localize there. This is supported by the abundance of bright green surrounding the nuclei, depicting a strong overlap between the D1 and mitochakra green localizations. One important note to make regarding the NIH3T3 cell line is that these cells in this image do not look particularly healthy. They should be more stretched out and elongated. A fresh population of 3T3 cells from the same original source were obtained from the more recent MTT assay. The three graphs here show the growth curves created from the trypan blue exclusion assay, where viable cells were manually counted at 6, 24, 48, and 72 hours of treatment. In Figure B, the A549 cells underwent an additional trypan blue assay, where cell counts were also taken at 2-hour intervals between 24 and 48 hours of treatment, in addition to the normal 6 and 72 hours. The blue symbols indicate the control, where no D1 was added. The orange symbols indicate 60 nanograms per ml of D1 treatment, and gray shows 100 nanograms per ml of D1. In general, across all three cell lines, the higher concentration of D1 reduced growth the most. As seen in Figure A, while the non-cancerous 3T3 cells were not as severely impacted by the D1 compared to the cancer cell lines, their growth was significantly reduced. This is hypothesized to be a result of testing with a non-healthy cell line as previously mentioned. The human lung adenocarcinoma cells in Figure B appeared to be the most affected by the dye. Interestingly, in Figure C for the T24 line, growth was noticeably reduced until an unexpected surge at 72 hours. This exponential growth is also present in the T24 cells not treated with D1, indicating that the surge in numbers may be related to its normal population growth. These two graphs are the growth curves which resulted from the PsiQuant MTT cell viability assay, where relative growth was measured at 24, 48, and 72 hours of treatment using a plate reader. The color scheme for the control, 60, and 100 nanograms per ml treatment remain as blue, orange, and gray respectively. In addition, the R-squared values for the exponential trend lines are at 0.96 or above, which is a significant improvement from the previous manual cell counting. In addition, this assay was conducted after a new NIH3T3 cell population was obtained. As seen in Figure A, these non-cancerous cells were less affected by the D1 treatment, showing a much smaller reduction in cell growth at 72 hours compared to the previous assay. Figure B also supports the susceptibility of the A549 lung adenocarcinoma cells against the D1. However, only a significant reduction in growth is present in the 100 nanogram per ml treatment after 72 hours. The MTT assay for the T24 urinary bladder carcinoma cell line has not yet been conducted. The selectivity ratios of the control dye D1 indicate a relatively insignificant selectivity compared to the other dyes of the same cyanide family. Cytotoxicity values were first calculated as a % reduction of cells between treatments and controls within each cell line at each time point. Selectivity was finally calculated as a ratio between % reduction in cancerous versus non-cancerous cells at each time point for 60 and 100 nanogram per ml D1 treatments. These ratios are detailed in the graphs on the screen. Figure A shows selectivity ratios of T24 human urinary bladder carcinoma cells from the trypan blue exclusion assay. Figure B depicts the ratios of A549 human lung adenocarcinoma cells from the same assay, while Figure C shows the results from the A549 cells in the MTT assay. Ratios below 1 indicate that more non-cancerous control cells were killed than cancerous ones. This is seen in the second half of treatment for the T24 cells in Figure A, and for the first half of treatment in the A549 cells in Figure C. What is interesting to note is the seemingly contradictory information between the trypan blue exclusion and the MTT assays. While the overall selectivity of D1 towards A549 cells decreases over time in Figure B, the selectivity is actually shown to increase over time for the same cell line in Figure C, which used the MTT assay. Currently, it is unknown why the results oppose each other. However, a future MTT assay on the T24 cell line may yield some additional information. None of the observed selectivity ratios reached above 3 to 1, a value which pales in comparison to the known potential of the other cyanide dyes depicted at the beginning of this presentation. As a control familial dye, a comparison of this D1 compound to the other cyanide dye being manufactured may provide some insight into what makes some family members possess extraordinary anti-cancer capabilities. My future work entails expanding the testing of our control dye to more cancer cell lines, as well as adding a new control cell line, CV1, which was used in the initial publication of the cyanide dye group's anti-cancer properties. Once manufactured, the other familial dye will be tested in an identical fashion as D1. In addition, the biocompatibility of various drug delivery vehicles will be investigated. I would like to take this time to thank those who have supported this research project.